with Elizabeth and Peter Provost from the OBIS Secretariat. So we're looking at how we're tackling biodiversity data provenance in this session, why it's important to know where our data come from and how it's been used and changed along its journey. But it isn't always easy. There's technical difficulties, inconsistent identifiers, poor interoperability, but also cultural ones like differing views on open data and attribution. So we've got some great talks in this session, some really good examples of how the community are working together to tackle some of these issues. We've got the microphone here, which is tiny but clips on, and although it isn't amplifying, it is being used for the online translation. So if I can ask all the speakers to use this. Also, if there's questions, we'll be handing this around, so one of us will be running around with the microphone for questions. So we're going to run through our talks. At the end of each talk, there'll be opportunity for 1 or 2 questions, and then time allowing, we'll open up the floor for questions for all our speakers at the end of the session. So without any further ado, I'd like to invite Ken Bagstad up to present our first talk today, looking at leveraging AI-integrated semantic modeling technology. Ken, thank you.
Thanks so much. Good afternoon, and on behalf of my co-authors...
Oh, there's no presentation on that one.
How do I just say next slide?
Yeah. Oh, sorry, yeah, next slide. There's no...
Perfect, okay. Actually, let's go back to the first slide. On behalf of my co-authors, I'm happy to talk today on why we believe that semantic AI is an important yet generally overlooked piece of the puzzle in our global biodiversity reporting systems. Next slide. So all of us here, of course, have an interest in supporting the kind of monitoring and reporting systems shown there on the left, and there are a number of technologies for modern data science that have emerged in recent years. And yet I think many of us appreciate that although these technologies are important and their adoption is increasing, they aren't yet enough to manage this very siloed and fragmented data landscape that we constantly hear about at this conference. Next. So I'm going to talk today about work I've been involved with for close to 20 years on a project called ARIES, Artificial Intelligence for Environment and Sustainability. It uses semantics and machine reasoning. So this isn't the machine learning-based AI that you hear about all the time, although we do use ML for certain applications. But I'm going to talk about some of the work we've been doing to bridge this space and to try to provide solutions. We are working on semantics that are able to cross disciplines. That's a piece I think is really important that I'll talk about in a moment. A federated approach to data and models we've heard about in many other sessions. The use of machine reasoning, which I'll describe in a moment, to manage the data and models. And a digital twin approach that can monitor the surrounding data landscape and provide updates when new data sources arise. Next. So we hear sometimes about the importance of discoverability and accessibility, and I think that's true, but I think we need to go a step beyond those to really machine actionability and machine understandability and interpretability of data. And so the way we do this is we start with semantics that are meant to be understandable and definable by a human and yet easily parsed by machines as well. And there's an example of that at the bottom. And one of the things that we really need to do is to make sure that those semantics are very modular and atomic, because if we have just a list of terms, say from a vocabulary, and we keep building onto that, that can get very unwieldy, and it's really hard to maintain the logical consistency that a machine needs to be able to make heads or tails of how these various concepts are related to each other. Next. The other piece that we feel is very important is to be disciplinarily agnostic, and so many different subfields, even within the biodiversity space, of course, are coming up with their own semantics and vocabularies, and that's great. They serve an important purpose for those communities to standardize metadata and reporting. But if we think about making this work across the scientific fields, and even for biodiversity, which is going to be composed of many different applications. For ecosystem services, the space I work in, it's very interdisciplinary, and so we have to sort of give up our attraction to these disciplinary terms and choose something that we call a semantic glue that lets us use a common sort of ontology for fundamental scientific measurements reuse existing vocabularies where we can, because there's lots of good work and there's no need to duplicate it, but also to be able to crosswalk back to that discipline-specific terminology. Next. The last piece of the puzzle here is machine reasoning, which I alluded to earlier. And this is designed for a machine to be able to for a particular problem that it's given by a human, to be able to look at what are the various models that could solve that problem, which ones are most appropriate for that area of interest, based on things like fit for purpose, spatiotemporal match, local specificity, and user-defined quality metrics, and to be able to match the best available model to the problem of interest, and then to match the best available data sets to go with that model, to execute and run that model, to provide provenance that's transparent. And so we make our data and models, instead of static repositories on the web, make them dynamic and able to interact with each other. Next. This is my last slide. It's important to point out that other communities have done this, communities like genomics, the Internet of Things, the climate and agricultural model intercomparison projects have really come together as a community to define common standards. That's complex to do for biodiversity, but I believe it's critical. And I encourage you to learn more and to join us in that journey. This QR codes to a paper that I was the lead author on earlier in this year that talks about how we can build this kind of community. So thanks so much.
Thank you, Ken, for a lot of information in 5 minutes. That was fantastic. Do we have any questions in the room? We have opportunity now for questions, or you can ponder on the presentations and we'll open the floor at the end as well. Any questions online as well? No? Obviously very clear to everyone, conversations. So then, in which case, our next speaker is a virtual presentation from Katrina Exter from the Flanders Marine Institute. Katrina is going to be presenting on a model for the provenance of marine biological samples and data. So if we're able to play that presentation.
Given the name of the session, I don't think I really need to convince anyone that marines provide provenance. Marine biological data and observations arise from samples collected from the sea. A lot of these data, and the conclusions drawn from those data, are used to inform the state of the marine environment, with the hope that they will be used to encourage authorities and society into actions that will improve that environment. As such, it is super important that the data and conclusions therefrom are associated with provenance.
There's no questions.
This provenance should provide traceability. This is not about the correctness of the scientific conclusions, but rather about whether I can believe that the data arrive from properly collected and processed material and trustworthy calculations. It should provide traceability and transparency. Can I know who did and what was used in the processing? So if I have questions or issues, I can trace back to the stage in the work I need to inquire about. And thirdly, it should provide repeatability. It is so important that an experiment can be repeated and redone, either in exactly the same way to check on the results, or in a slightly different way to carry out different investigations. So what is the provenance information that should be provided? This is largely about who, what, where, when, and how. Who did the work? What are the samples and what was used to collect, store, process the samples and the data? Where and when did the samples come from and did the work take place? And how were the samples and the data obtained and processed? How should provenance be provided? Provenance should be provided as metadata. Now, metadata are just data that describe other data. metadata can provide many different types of descriptions: dataset discovery, technical details, usage information, and of course, provenance. Now, all data that are published in catalogs have discovery metadata: title, citation, author, date, locations. Provenance metadata will include some of this same information, dates and locations, for example, but it should include more, and it serves a different purpose. It's not for discovery, but it's to provide trustability, traceability, and reusability. Provenance metadata should therefore be provided as a standalone document that is accessible together with the dataset itself. The provenance metadata should describe only the data that it is linked to, not any preceding data used as input. However, it must include links to those preceding data and or material samples. Provenance metadata, to be equally understandable or equally readable by all, should follow a template or model. These templates or models will of course be applicable to the type of science, type of experiment, data that is described. So there will need to be community input to what information is necessary in provenance metadata for different domains. The metadata should be semantically annotated. That is, one should use terms from controlled vocabularies rather than words you make up yourself. So the same concept is written always in the same way and can be understood in the same way. Now, the metadata should clearly be readable by humans, but ideally they should also be readable and understandable by machines. That means software, search engines, by automated processes. This is because these machines are becoming almost more important than individual human scientists when it comes to finding, accessing, and using data to produce science. Okay, so on to our actual provenance model. We developed this model to fill a gap in the provision of provenance information that comes with most marine data sets that are published. Now, while most data catalogs do include metadata on temporal and geographical scope, coverage in terms of taxonomy and measurements, and sometimes also protocols, these fields are not always mandatory, and they can be filled with information that's not always directly accessible. For example, a protocol by name, rather than a web address from where one can read. In addition, this provenance information does not always allow for traceability. You don't necessarily know what stage the protocols or instruments mentioned were used, for example. And information about storage or shipping and some other aspects of provenance are rarely provided. To address this shortcoming, and because of the time we started this work, we were involved in a provenance work package of a European Horizon project, EOS Live, we created a model for provenance for biological material, observations, and data, applicable in the first instance to the marine sciences. Now, our model does not contain anything really new. We use already existing standards, terms, and accommodations in the data world for provenance. It's in the stitching it together, our use case, and in our implementations, that we are doing something new. So this is our model. It consists of the following classes: activities, agents, locations, and entities. Where each class has properties that allow one to describe the activity or entity, to link those to the responsible agent and location, and to make the activity, entity, agent, or location globally and uniquely identifiable. Now, this last point is important. For the provenance metadata to be fully machine findable, and for one provenance description not to be confused with another one, when all the metadata are incorporated in an object, such as maintained by search engines like Google, but also Ocean Info Hub, it is important that items of provenance metadata can be made globally and uniquely identifiable. Now, this is, to be fair, an advanced use case for a model, but it's one that we at Wilson and the MLC find to be important. So this is a diagram of the classes of the model that are entities on the left and agents and locations on the right. Each box here is a class with the properties of each class listed within the boxes. I'm not going to take you through all of this, but to pick out some highlights. In our entities, we account for platforms and sampling and observing devices, samples, protocols, permits, and software, whether standalone or as used within a device. Each class has its own properties. Those in common to all are listed in the top entity. Agents can be people or organizations, and locations can be any geographical point or shape or just an address. The activities, we split them into two, digital and physical. Digital are data processing and acquisition of data, for example, using published data to create new data. Physical includes sampling and observing, acquiring material, transporting and storing, including biobanking and processing. The properties in common to all are listed in the activity box at the top. Properties of all the classes include the agents, locations, and entities that we showed in the previous slide. Okay, so all well and good, but the proof is in the eating of the pudding, as the saying goes. How can this be implemented? We are still in the process of fine-tuning the model, so we don't have any actual implementations just yet. However, we have turned the conceptual model, classes and properties that I just showed you, into templates in two machine-readable and understandable formats, JSON-LD and EML, ecological metadata language, which underpins OBIS and metadata. In addition, in order to demonstrate the model, we've created a long but realistic example of a marine biology experiment which produces multiple data sets. For each of these data sets, we've created an associated provenance metadata file in JSON-LD for machines and in plain text for humans. This is the example experiment and its three data sets. It's a sampling event producing log sheets, plankton species identifications from images, and sequences from samples. While our templates and examples can be found on our GitHub site, you can see them here, although please do note that this is a work in progress, so the files in GitHub are still not final. Okay, so we aim to publish this work early next year. Meanwhile, we are implementing it in EMBRC's IMABORN project, where taxonomic lists, measurements, and sequences obtained from IMABORN samples will all be linked to their own provenance trail. Now, in IMABORN, we already collect some 90% of the provenance information in our extended log sheets. The hard part is about putting it together in a way that will allow us to store that metadata efficiently and effectively so that anyone can ask for any part of the IMABORN data and get a provenance metadata file that is unique to that part of the data. In addition, parts of the model are being incorporated in work we are doing in another European Horizon project called Marco Polo. And finally, we will provide templates and documentation as part of various fair data courses that we run at LIS and for EMBRC. Thank you for your attention.
Thank you. So Katrina is online, but obviously she's able to respond by typing rather than speaking. So if we do have any questions for Katrina, we can speak them now. She'll type them out. And we'll put them in at the end of the session. We'll feed back her response. So, any questions in the room, Elizabeth?
Yeah, I was just wondering, the GitHub link to where all the templates were being produced, are these the same or similar templates that ODIS has been producing? I didn't quite see the full link, but it looked different from the repository that I know that ODIS is putting their templates. So I was just wondering if they're the same or different.
So we'll have the delayed response from Katrina. So yeah, come back later for the response. It keeps you all in room and engaged. Any other questions? We have got a few questions coming in online for the previous presenter, for Ken as well, but we'll return to those at the end of the session. In which case, we will move on to our next in-person presentation from Jonas Grieb from the Senckenberg Institute. He's going to be talking about tracking natural science objects and their physical and digital derivatives.
Yeah, good afternoon. and thank you for the opportunity to present this topic today. All of this presentation and this work would not have been possible without valuable contributions from my co-authors, namely James, David, Christian, Michaela, Etta, Volker, and Klaus. Next slide, please. So I'm going to talk about natural science objects in the sense of material samples and physical specimens, which can be considered knowledge archives of life, which conserve snapshots of biodiversity. These objects are typically stored either at natural history museums or at related environmental research institutions. For example, the Senckenberg, where I work, we are a big natural history museum in Germany. We have over 45 million preserved specimens from all types of disciplines: botany, entomology, mammalogy, both recent and fossil collections. Then there are also these environmental samples, so soil samples or water samples, which may contain organism material, and these even become more and more important in the context of eDNA research. Now, both material samples and physical specimens have in common that these are actually dynamic entities. They are not static. They undergo several preservation and analysis processes, often even including subsampling, which really means altering the original object physically, and for example, by splitting it up in numerous smaller samples. Additionally, metadata, the information we have recorded about a sample, is continuously refined with new evidence, and all of these processes need to be preserved, and the provenance needs to be tracked. However, collection management systems traditionally do not handle this part very well. They struggle especially with modeling complex relationships, like when we create subsamples, we have relationships between different material samples, and all the history that needs to be recorded, typically is an issue. Now, because of this issue and because of some other data management requirements, we created DINA. Next slide, please. DINA is the name of a collection management system software and also the name of a consortium behind it, a consortium that was founded in 2014 by natural history institutions from Europe and North America. You can see the logos of the institutions there on this slide. And it was founded with the aim to build an open and collaborative collection management framework to support specimen and sample-based research. This is also based on past experiences in many institutions where we tried to build our own in-house solutions for databases, and this was often a dead end at some point, and such a collaboratively developed open source software promises long-term maintainability. DINA is governed by an international steering committee, a technical steering committee. However, I want to talk now more about how this software actually allows to model this provenance. And for this, we need to talk about DINA's underlying data model that was also collaboratively designed with the aim to be both flexible and really model complex relationships between samples. And therefore, in the following, I will present 3 examples of, let's say, real-life trajectories of physical specimens. And all those trajectories and the relationships, as shown, can be actually modeled in a machine-readable way in DINA. Next slide, please. The first example is from a fossil collection from an amber object at Senckenberg. Those amber objects usually enter the collection. They are assigned a catalog number. They bring some provenance information with them, where they are collected and so on. Possibly a photo is taken. This photo can also be stored in DINA in the system. Is linked to the original object. We establish this digital derivative relationship. However, special for Ember objects is that they often contain organic material from more than one individual. And yeah, because of this, they are often sawed up into smaller pieces, which is a process that one can consider subsampling. So in this example, there is a tiny subsample created. Actually, all of the subsamples are created as entities in DINA. They receive their unique identifiers. In this case, a small portion is sent to a laboratory for some chemical analysis. Then we might have another subsample containing exactly one insect of interest, so a certain individual, which is then digitized. For example, there's a 3D model created. Again, we store this 3D model, create this digital derivative relationship, and at the end, yeah, the subsamples are stored, sometimes also shared, exchanged with other museums. So it's quite easy to lose information, but in DINA we always have these links back to the parent material sample and thus maintain the provenance information. Next slide, please. The second example is to illustrate that provenance information has also a regulatory aspect. So in this example, we can consider the leftovers of a spectacled bear species that's actually present here in this part of South America. If we imagine that some, let's say, physical remains of such an individual are collected here, possibly transported to a research institution in another country, then the Nagoya Protocol applies. So we have regulatory documentations, for example, a material transfer agreement. This can also be, let's say, stored in DINA in the so-called object store module. It can be linked to the original collecting event of the physical sample. Then again, we might have some subsampling process, because for mammal collections it's typical to have bones or teeth stored separately, then tissues go in a specialized tissue collection. There might be, again, a subsampling process. In the end, we extract some DNA information, and again, everything can be linked back, traced back to the original physical sample, and thus also to any regulatory documents attached. Next slide, please. This final example shows a more environmental sample. It's a soil core sample that is collected. We have again some collecting event information, possibly also a material transfer agreement. And again, we create subsamples also in DINA. This can be modeled, linked back to the original sample, and everything can be traced and linked until the machine identifications of the contained eDNA in the sample. And next slide, please. Now I talked a lot about DINA's data model. There are also some other aspects. We try to promote provenance and standardizations. For example, with respect to determinations in the DINA's collection management system, it supports or even endorses that determinations, scientific names, are retrieved from global taxonomic backbones, and they are linked via their persistent identifiers, and this is always preferred over building taxonomies internal to the system. In the same way, DINA also integrates with other external data and databases, for agents, for geographic place names, for expeditions, always via their persistent identifiers. And finally, also to mention that DINA, since the beginning on, always had an API, so data stored in DINA can also be integrated by other systems. Next slide, please. The outlook, what we want to do with the DINA collection management system. So first we want to explore the serialization into the upcoming Darwin Core data package that was extensively discussed already at this conference, and we believe that there is already a lot of alignment on the conceptual level. At the moment, we're working on implementing the management of expeditions and sites in DINA so that you can link collecting events to this. We're also building or working on further integration on specific data fields for Earth science data objects, for geological collections, and so on. And finally, we want to build dashboards to show this complex derivative and relationship between material samples. So it can already be modeled in DINA, but let's say dashboards would make these relationships much better accessible to the end user. And next slide. There was one slide. Yeah. With this, I want to acknowledge a colleague from Senckenberg for the nice amber photos, and yeah, I want to acknowledge all collaborators who worked on the DINA data model. And finally, I also want to invite you, if you're intrigued by DINA's approach, by DINA's data model, and if you have to manage physical samples, we're an open consortium. Feel free to approach me or approach James, who's there. And yeah, let's get in touch, and thanks for the attention.
Thank you. As always, we have opportunity for a couple of questions from the room. Any online questions we will hold till the end of the session. Yep, question at the back, Catherine.
This is the smallest microphone I've ever seen. Does it even work? Great. I'm impressed. Thank you for your talk. I work for CSIRO in Australia, and we manage samples that are collected on the nation's blue water research vessel. Some of those are sediments, some of them are rocks, some of them are biological, and we don't have a sample track. We don't track sample provenance, and we need to. My go-to is going to be to consider IGSN as our first port of call, and I wonder if you have any experience with IGSN and can tell me, how they compare, how it compares with DINA, especially in terms of the fish catch and the biological samples that we're collecting.
So it's something that we have not implemented yet. I could imagine that we come to this in the future. So in the end, DINA is a software that you would run by yourself. It's like a self-hosted repository, which also some institutions might prefer to have their data really in their own infrastructure. And then we can definitely work on integrating that samples created in there. Let's say you mint IGSN identifiers for this, but it's not done yet.
Thank you, James. Okay, we shall move on to our next presentation, which is another in-person presentation from Elizabeth Lawrence from the OBIS team. She's going to be talking about how she's simplifying metadata flows with an essential ocean variable application.
Yeah, great. Thank you. Okay, so before I get started, I just want to acknowledge the work that I've been doing to develop this app has been supported by the Horizon Europe project BioEcoOcean. So thank you for that. And next slide. So I'm sure all of us in the room are familiar with what FAIR means, but very quickly I just want to go over each of the aspects of FAIR. So next. So findable, of course, means that there's descriptive metadata with our, that includes stable links, DOIs, and so on. So your data, your object, whatever it is very findable. Next. Accessible means that there's very clear instructions on how your digital object can be accessed, and this goes beyond just a available upon request statement. Next. Interoperable means that your data, specifically for alert referring to data sets, are easily combined with other data sets and are, importantly, as the other talks have really made that clear, are machine readable. And so this usually means that using data standards and controlled vocabulary that is both human and machine readable. Next. And finally, reusable, making sure that we have metadata that really enables replication. So it's descriptive enough that if you're looking at your own thing from 5, 10 years ago, 3 years ago, you know exactly what it was. Other people can also read your metadata and be able to replicate what was done. Next. So just mentioned metadata. The focus of my talk is about metadata. So next. Speaking of metadata, in the biological ocean observing world, it can take some time to get the biological data from, in situ. We are doing the observation into a digital publication. And but we still need to know what monitoring is being done where. And this is why metadata about who's doing the monitoring is really important. And this is captured by the Goose BioEco portal. So next. The Goose BioEco portal provides an interactive map that shows us exactly that. It shows us who is collecting data on, for example, biological and ecosystem essential ocean variables. Next. Where is the sampling being done? Next. And it also provides extra descriptive metadata about the monitoring efforts. Next. And I just wanted to point out that while the URL for the BioEco portal is at the top, we are working to redevelop it that will use linked data to be able to really make it more interoperable. And this is a work in progress with colleagues, Obis in the room, Peter. Next, please. So speaking of linked data, within the IOC UNESCO digital ecosystem, this is where a lot of the work for linked data in the ocean observing world is being done. So we have Obis, ODIS, and the Ocean Info Hub. So Katrina, I believe she mentioned Ocean Info Hub, and Goose, the BioEco portal. So all of these aspects are part of the IOC UNESCO and are working to develop interoperable linked data standards. Next. So taking a little closer look at ODIS, this is the Ocean Data and Information System. So ODIS is not a repository, but it's rather a federated system that lays the infrastructure and provides metadata schemas using schema.org vocabulary and JSON-LD format. Again, Katrina was talking about this, which is really, really great that she gave that foundation. And what's been pretty cool about this is the templates being developed have feedback from the community. So ODIS is really working to make resources more discoverable, as I said, using linked web architecture and linked data. And so all the metadata templates are in JSON-LD. So next slide. So this is what a JSON-LD file looks like, which I think most of us in the room are probably pretty technically savvy. That might not be the most intimidating, but next. For any non-technical users, they're going to look at this and feel very intimidated and ask, how am I supposed to create my metadata in this format? And that can be very discouraging. And of course, we don't want to discourage our biological observing community. We want them to feel empowered to share their metadata. Next slide. So this has led to the development of a metadata application that will facilitate and make it a lot easier for non-technical users to fill in their metadata about their ocean observing program. So the way this app works, next, it has a login. You log in through your GitHub account. You fill in, it's just a very user-friendly metadata form. Next. And then in the back end, it will take the form and produce the JSON LD file for you. Next. And then that JSON LD file is then pushed and saved on a GitHub repository. And sitemaps are also created so that way that it all automatically connects with ODIS and the BioEco portal. And so just by entering, a user can enter in the form, and that describes their ocean observing. And they don't have to worry about the back end. If they want to, they can go look and take a look at the JSON LD file. And we're also ensuring all of the metadata captured aligns with the essential ocean variable specification sheets. So making sure we're capturing, like using the same keywords, using the same terminology, and so on. Next. So this is a little screenshot of the form. I can't remember if I might have put a little video, but yeah, so the metadata form, it includes spots for you to put an identifier for your program, your project, your monitoring program. And if you don't already have an identifier, we can create a DOI for you. So that way we can reuse these identifiers in, for example, OBIS data sets. And then everything will be linked. You can see data sets produced by the same project that has the same identifier. It also uses keywords from other like interoperable vocabulary servers, so like marine regions for geographic areas, and like the biodiversity, NERC, other control vocabulary for keywords. Yeah, including EOBs. Next. There is a video. Okay, we can skip that. It's okay. Some of the upcoming work that we're working on is some additional data standardization tools that can kind of help facilitate converting like EOV format data into Darwin Core. And I do have a QR code at the top that goes to the app. I will say that a few weeks before the conference, we had to do a little server update, and so you can't actually submit anything right now, but you can still check it out, and hopefully right after the conference we'll get that sorted ASAP. Next slide. Yes. Great. So that is the end of my talk. And again, thank you for my funders, BioEco Ocean, and the European Commission, European Union, through the Horizon Europe project. Happy to take questions. A bit short, yeah.
You can ask a question. You're a co-winner.
Yes. Ask myself a question.
What feedback have you gotten from your users on that user metadata generation form, and how are you incorporating it?
So this is still a pretty early development, but most of the feedback so far has been, especially from members of the BioEco Ocean project. And when I first showed them the JSON LD file, very shocked and overwhelmed, I would say, was the response. And then when I showed them the form, they're like, like this is great, this is fantastic. Can we add this? Can we capture that? Oh, can we add some clarification about what this thing means? So that's mostly the main feedback has been around what else can we capture in the form so far. And it's nice.
Compared to JSON LD, yeah. Great. Thanks, Elizabeth. Yep, question?
Is this what? Can you share maybe what things they wanted added or clarification on the form?
So some of the things that I was requested to add initially was including a list of like EBVs, so the essential biodiversity variables, and adding like little info bubble to describe, like, trying to remember an exact. I use the word data producer to describe like any type of ocean monitoring, because if I've heard that we used to use the word like, oh, program, and then users were like, oh, well, I'm not running a program, I'm just a PI doing long-term monitoring. And so I kind of generalize it to data producer, but then of course, we need to describe what that is. So, and there's some others, I can't think of them at the moment. Yeah.
Okay, our next presenter is Dekai Kao from the Taiwan Biodiversity Information Facility. This is a virtual presentation speaking on embedding provenance into biodiversity data.
Hello everyone. Today I'm here to share our experience with Taiwan's long-term social ecological research platform, focusing on the theme of trust, traceability, and transparency in data provenance. My name is Dekai Kao, you can also call me Derek, and I work on data administration at Taipei. Data provenance is essential for research, policymaking, and long-term sustainability. But it's not only a technical issue, it also depends on human-centered collaboration and long-term institutional support. In this talk, I'll first explain why provenance matters, and then show how we put it into practice. In Taiwan's LTSR, we work with very diverse data: ecological monitoring, species observations, and even sociocultural information. Because of this heterogeneity, seamless integration across domains, the question becomes, how do we bring all these data sets into a common framework of traceability and transparency? Let me walk you through the workflow here. Contributors first provide very diverse data sets. To support them, we built an AI assistant called Digital Typing. It works like a GPT chatbot, but with Darwin core documents attached to the background. In other words, it uses a retrieval augmented generation RAG approach, so contributors can just ask questions in natural language and get guidance on field names and standards in real time. After that, the data goes into the validation. Here there are really two possible paths. Option one, I handle the conversion myself. I write Python scripts on Google Colab and contributors just click through the steps. Option two, we emphasize education. Contributors learn the standards and use the cleaning tools themselves. Once the data is ready, everything flows in the depositor platform. Each data set receives a persistent ARC ID, follows FAIR principles, and can be accessed or visualized through the API. And most importantly, you see that red arrow in the center of the diagram. Requires ongoing collaboration. So, how do we actually deal with these challenges? I actually took over this role from a previous data manager. He had already worked with contributors to agree on certain standards, but in practice, contributors often don't follow the standards. Sometimes they forget, sometimes they misremember the version. And when this happens, it's tempting to argue, why didn't you follow what we agreed? But I've also been a field researcher myself. I know how exhausting it is to climb mountains or dive into the ocean to collect data. At that point, asking someone to redo their data entry feels frustrating and discouraging. There are really two approaches I can take, but each comes with different costs. Option one is that I understand the raw data, and then I write a Python script to convert them into the relevant standards, such as Darwin Core. I usually implement this on Google Colab, so the contributors just click Next Step and get clean data. In this case, almost all the cost is on me. I spend time writing and maintaining the code, but the advantage is that the cleaning process is transparent. I keep versions in Google Drive, so provenance is preserved in the code history. Option two is more about education. I not only have to understand their raw data, but also explain what these standards are, why we use them, and how to do the mapping. Then I teach them how to use the cleaning tools. This builds awareness, but the ongoing cost is very high for both them and me. And at TaiBIF, our mission is to promote open data and the adoption of standards. But whether contributors are willing to learn and apply them really depends on their own mindset. And that's something beyond my control. So while we all recognize the importance of capacity building,
In practice, the cost effectiveness is very different between the two options. Here's our takeaway. Provenance only works with continuous engagement, not a one-time agreement. From the two paths, option 1 reliably delivers this batch with a reproducible evidence trail, while option 2 builds capacity over time. Our next step is to pilot Clean Then Teach. We clean this batch via script, then teach using their own before-after, hand over a checklist, and keep a light review loop. After data are standardized, the next step is to make sure they're preserved and accessible in the long term. That's where the depositar platform comes in. Each dataset receives a persistent ARC ID and follows FAIR principles. With the API, datasets can be uploaded, accessed, and visualized directly on project websites. This way, provenance is preserved not only during cleaning, but across the entire data lifecycle. Thank you, everyone. If you have any questions, please feel free to post them on Slido. I'll make sure to reply. For direct contact, you can also reach me at plant58697@gmail.com.
Posted a Slido as well that he will respond to, but we can also ask questions here and have him respond, and we'll feed them back at the end of this session. Okay. In that case, our next speaker is Edward Gilbert from the University of Kansas, talking about enhancing provenance and interoperability in biodiversity informatics. Over to you.
All right, I'm gonna so I'm gonna quickly walk through how data provenance and interoperability can be enhanced through a decentralized network of Symbiota data portals. Next slide. So Symbiota is an open source software that's specifically designed to build biodiversity data portals that centers around specimen data. And so Symbiota, there's no data associated with it, it's just a software, but if a project wants to establish a biodiversity portal, they could download the software, adjust the configuration files, load data, and it gives it a particular look and feel that represents their research group. So next. On the left here, there's a bunch of portals, example portals. Next, you can see they all have a different taxonomic and geographic scope associated with them. Next. And the most successful portals are the ones that are run and governed by a collection, a consortium of biological collections. Next. And Symbiota, it's a content management system. Next. So all the data in it could be modified directly through the user interfaces. Next. And next slide. So this slide shows you the basic workflow that exposes our data that the community has accepted. So at the bottom row, that's all the collections. They're being managed within different software. Ideally, we have a well-designed workflow within the community to get the data from the source data up to the data aggregators at the top, such as GBIF there. Next. And so flow up is good. The researchers, they'll find the data at GBIF, and for instance, maybe someone wants to do species distribution modeling, they'll download a subset of the data to a local system, they'll do some data cleaning, they might fix coordinates, add coordinates, resolve taxonomic discrepancies. This is all done locally. Next. So the little arrow going down is there's not a good infrastructure to get those improved data annotations down to the source data. And next, I'm going to focus on this talk. I'm going to focus in the middle area. That's the Symbiota portals. Symbiota portals are kind of like a data aggregator, such as the top, but also data management thing at the bottom. Next. So each of these little squares or circles represent a symbiotic portal. And some of the portals, such as the one in the bottom center, the California Consortium portal, are dedicated to providing support for collection management directly within the portal for a collection. So each of those little green circles is a different collection, managing the data directly within the portal. However, they usually want to publish their data out to data aggregators, such as GBIF, but also to other symbiotic portals. So they'll push out their data. There's a well-designed data publishing tool built into Symbiota that acts like an IPT that allows them to push out subsets of the data to the Guatemala, or the Gabonese data to the Gabon. Next. However, traditionally, we struggled within the Symbiota network to get annotations that are made on snapshot records back down to the source collection. So it's the same problem that was demonstrated in the previous slide. Next. So we built in, over the last 2 years, we've been focusing on building in 4 levels of infrastructure to help solve this problem within the Symbiota network, so that there's more bidirectional data flow between the portals. So the first part is more robust API. So that's a second version of the API built into Symbiota. The second part is a decentralized portal network. So if you establish a new portal, you could reach out to the other Symbiota portals, and within minutes, they will understand where you are, that you exist, and you'll understand where all the other Symbiota portals exist. And then the ability to track data provenance on a record-to-record scale. So when you push out data, snapshot records out, you know where all your snapshot records are, and you also, the snapshots, they know where they came from. And then ability data versioning on a record scale, where you know who edited what the old value is, when it was edited, and what the new value is. Next, this is my last slide. So through this infrastructure, now we have a situation where you have your live managed records within a Symbiota portal. You push them out. Now, if there's an annotation made on a record, that annotation is immediately pushed to all the snapshots. So now the snapshot records are synchronized with the live managed records. And then with the snapshot records, if an annotation is made on one of those snapshots, the collection manager can be notified of those annotations, and they could selectively pull that record, those annotations back to the source record. So now we have a situation in software where the researchers could download, install a Symbiota portal, pull down their data, do all these annotation things, and there's infrastructure to push the data back down to the source collection. That's it. And that's a Symbiota team that's working to build all this. Yep.
Do we have any questions for Edwin about Symbiota? No, obviously. Very clear, thank you. Oh, yeah, please.
So during the conference, there has also been comments that at GBIF level that they could add these corrections, right? So they're talking about that. So how do you see the timeline to pulling back corrections that may be suggested through the GBIF platform? Yeah.
That's something that we're very interested in doing, be able to pull the data back, and through the API, data could be pushed in with authentication. But this aspect is doesn't have the infrastructure to pull it down, but we are interested in doing that.
Thank you. Next presentation is online, and the speaker is also online, Joet Poland from University of California, Santa Barbara. And the presentation is on bookbinding for the digital age.
Hi, I'm Joet Poelen, and I'd like to talk to you about bookbinding for the digital age and describe a format-agnostic method to review and archive biodiversity ever since the internet became popular, we realized that the content published on it was ephemeral. Here's a quote from the Internet Archive mission statement. More recently, we've done a study on digital biodiversity data across known networks change or were unavailable in 2019 and 2020. So this begs the question, who or what keeps our digital biodiversity data around? The obvious question is, the data fairy. So who is this data fairy? Is GBIF our data fairy? According to their terms of use, GBIF is provides a publication framework, but is not the owner or the custodian of the data hosted in it. They also don't guarantee uninterrupted data services. So no, the GBIF is not our data fairy. They don't claim to be. Turn out that the data fairy is a cousin of the poop fairy. As in, there's no such thing. Recent reminders of the data fairy absences are, the 54 hosted collections that were affected by a infrastructure outage in hosted Symbiota portals. The Symbiota support team worked very hard to get it back up, but they got no help from the data fairies. Also, this big network of arthropod data was taken offline this year with no plans to revive it. And the Biodiversity Heritage Library, after digitizing over 300,000 physical biodiversity works, faces an uncertain future as they're set to lose their institutional sponsor next year. So in my mind, the thing that keeps our digital biodiversity data around is our awareness of how fragile this digital data is. And I'll talk a little bit about other things we can do to keep it around. Common sense data archiving and citation practices. So here's where I want to introduce this idea of digital bookbinding for biodiversity data. So what's so neat about physical books? So even before the invention of the book press, books and scrolls have been pretty successful, like transferring knowledge across generations around the world. They're just very simple things, a portable stack of bound paper containing text and images. They can be combined in collections without having to change their design, can be kept all over the place in public libraries or private collections. They're wireless, their content cannot be changed remotely. And if they are changed, it's pretty easy to detect them usually, like a ripped out page. And they don't need power to operate. They can be sent by mail. So here's an idea. Why don't we treat digital data more like a bound book instead of something hosted at a web location? So here's a recipe to binding digital data books. First, you collect the citations of the digital data that you want to use, and you make sure to use signed citations, securely linking to the data. Then you list this in what I call a data bill of materials, and also describe where they came from. Then you publish this data bill of materials, and this is just a digital text file, then you can use the citation to this bill of material in your research to point to the data collection that you use. Then last but not least, you'd want to make sure that this, this bill of material stays available, as well as the associated data contained in it. So here's an example of an actual bill of material for the IDIC biodata registry. I've been tracking the IDIC bio network for many years, and how it works is you look at the registry, and the registry contains links to institutional Dharma Core archive data URLs. You take a snapshot of both the registry and download the associated Dharma Core archives to create a bill of materials and the Dharma Core archive files. And you can do this by running this single command. So what does this look like, more or less? So expressing the bill of material in plain English would be something like, a version of the IDIC bio registry was downloaded on blah, blah, blah from this location, and we found this content. And then this specific content contained a data set with a certain identifier, and this data set was associated with, etc., etc. You can also take a more machine readable approach by using a provenance ontology and hash URIs in RDF enquads. Still text. So that looks a little bit like this. You have a URL that has a certain association with content at a certain point in time, and then this content contains some description of the data set. This data set is associated with, etc., etc. So as a text file, this bill of material has a content signature itself. And this content signature uniquely identifies the collection of data that it references inside of it. So in order to retrieve this collection, first you need to get the data bill of material by there, with a certain signature. So you can ask around, hey, do you have a bill of material with this signature, content signature? Then after you get and verify the bill of material, you can look at its contents and then look at the data citations in it and retrieve those. So note that we're asking for data content. We're not asking for specific data locations. We don't really care where the data comes from, as long as it's the content that we're asking for. Also, these content signatures are format agnostic. They can be any binary file of any size. So here's an actual example you can run on a bill of materials that includes the IDIC bio registry. And this lists the first record in the collection, which turned out to be a lizard specimen from Pakistan, hosted in the collection in Puerto Rico. So you can do similar things for instance, finding the last record. Like, what would that look like? You get the bill of materials. Here's the content signature for it. And then you can list the last record, which turned out to be a fungus. So this data bill of materials approach is pretty flexible. You can reference other data bill of materials, including the older versions of this one. They can be tiny or super huge, and they can include any arbitrary detailed description of the data it contains in either English, Spanish, or any other natural language or structured text or a combination of them. So with this, you have a recursive data structure that you can cite, and it can grow to an arbitrary size. Here's some more examples of existing data bill of materials. So this is a corpus from the Biodiversity Heritage Library. All the PDFs contained in it and their metadata. And then this is all the Darwin Core archives associated with the now defunct SCAN data collection network. So who or what keeps our digital biodiversity data around? I think it's the awareness of fragility of digital data and common sense data archiving and citation practices. I'm using bill of materials approach since September 2018, and I'm curious to learn about like how you intend to keep our digital heritage around. Thank you for listening, and remember, the data fairy does not exist. Thank you. Thank you.
Thank you, Joanne. I know Joanne's online. That's a fantastic message. I think we probably all need to remember that there is no magical data fairy. Brings us on. Are there any questions? Sorry for Joanne. Yeah.
Well, yeah, it was an interesting analogy to ponder, but the books did have or do have the fairy, the libraries. And so for me here was not really clear having the awareness of the fragility, but you are keeping, you know, copies in several places, but how do you see the library system working with your system?
Thanks, and we'll let Joanne respond and come back to it at the end of the session. Make sure we've got time to feed that back. I know he's active in the chat, so we will get an answer. Our final presentation today is another virtual one from Guillaume Body from CNRS in France. And he's going to be talking about the BioDash tool, which is used to describe essential biodiversity variables and a dashboard to describe them.
Of the French Biodiversity Agency and the National Museum of Natural History. And I'm going to present you BioDash, the EBV-based dashboard of Europe. There is a huge diversity in monitoring schemes across the different scales, and we are talking not about hundreds, as we identified in different reports in Europe and national level, but about thousands of monitoring schemes, and it's very hard to understand who is doing what and what do we know about biodiversity. We also identify that there is different actors that are interested by knowing this landscape of monitoring schemes. There is first the strategist. Their job is to manage monitoring programs that are composed of many different schemes. And their need is to have an overview of the landscape at their scale. And you have strategists at national, European, world, or even local scale. Then you have the scheme leaders, which is maybe the most important role. They have in charge one monitoring schemes, and they guarantee the science, the training of observers, the collection of data, and their analysis. And their need is to present their work and to obtain reward about their job. And remember, they are the only one providing information to the system. And then you have researchers. Their job is to reanalyze data for new knowledge, often combining different data sources and different monitoring schemes. Their need is to find data and understand how they have been collected. So we designed BioDash and different functionalities of BioDash. First, it must be able to describe monitoring schemes, their results, and how to access to their data. We also want to be able to research one particular monitoring schemes based on different criteria. We also want to have a synthesis about the monitoring landscape across all schemes to know what do we know about birds in France, for instance. We also want to improve the connection between monitoring schemes to know, for instance, that someone else is running monitoring which is very similar than yours, but in a different country, and you didn't know about it. And as we want to manage the systems, we need to know the costs and the resources needed to run monitoring schemes to ensure they are correctly funded. And also to reveal this funding landscape across all scales that are sometimes very hard to understand and to reveal. So we want to share information. We want to work across scales to provide services to the different users and to acknowledge the job which is well done by monitoring scheme leaders. So the BioDash project will ensure trust, traceability, and transparency in monitoring schemes by having access to descriptive monitoring scheme cards to their results using the EBV approach, to have a synthetic overview of the landscape, and to create a network between monitoring stakeholders. We work at European and national scale, and we focus on strategists, funders, data analysts, and monitoring scheme leaders, and we hope to have it running by 2027, and you will be able to use it across all the world if you want to. I thank you very much.
We won't take any questions unless I hear differently. So that brings us back to opening the floor for any other questions that might have occurred to you. For any of our presenters, we do have some questions online that I'm going to come to as well. But please raise your hands if you have any questions. The first one is a question for Ken. So it's from Katrina, one of our other presenters. Do you think it's possible to use AI reasoning to correct or add to historical metadata, e.g. where new metadata fields are added after the fact, or metadata are missing but could be harvested from related publications, etc.?
That's a great question, which I think I would want to approach very carefully, especially depending on how far back in history we're talking, how easy is it to reach the original authors. I didn't talk specifically about the use of large language models in helping to derive metadata. One of the speakers, the Taiwanese speaker, did talk a little bit about that. Obviously, there's a heavy human QA/QC component to doing so. But I think it's an interesting idea and an important one to explore.
Another question that's come on for you as well, from Jorit, who, again, one of our presenters. Question, in your Aries platform, how do you distinguish between the provenance of digital data, i.e. bits and bytes, versus the provenance of data records, information contained in the data? Repeat the question. Yeah. So in Aries, how do you distinguish between the provenance of the digital data, the bits and bytes of the data, versus the provenance of the data records that contained information?
The main we don't explicitly tackle the provenance of the original data sources. A lot of our work is in producing proper provenance for a workflow that involves modeling. So I think that's where some of the provenance schemes that some of the other presenters in this session talked about are really important. But when it comes to the workflow side of things, that we think is really important in any sort of modeling approach, which can include modeling of any type of interpolation or the sorts of things we do with species records to produce derived data sets. And that we do produce provenance for that shows the workflow, the steps taken, and provides a replicable pipeline.
Feel free, if anyone has questions that occur or sparked by the discussion, please raise your hands as well. Otherwise, I'm going to keep going through the online questions. Question for Ed Gilbert now. Can you talk a little about the adoption of your provenance versioning of records kept in Symbiota?
So we have tables that maintain the versioning of the records. So anytime anyone does an edit, it records the time, the person, and which fields are edited in old value and new value. So that way it keeps track of all the edits through times, but also we could use this to control the ability of pulling the edits back from snapshot records. And so there's data snapshots within the portal. If people are going to refresh those data snapshots, we don't want those edits to be copied over. So storing them within a separate table allows us to keep track of all that.
Thank you, and apologies, this is slightly sort of artificial and kind of stagnated conversation in the way that we have to deal with the online participants. Elizabeth had a question for Katrina, asking about the Marco Polo and ODIS data. So Katrina's responded. The work we're doing in Marco Polo is indeed with ODIS. The particular link she gave is for the full model, which is just for provenance, while the ODIS work is for all metadata, discovery, description, acknowledgement, including provenance. The two templates will therefore not be the same, but cross over in places. She also wants to add that you did learn some useful things from the work with ODIS in their RDF implementation of the model. So yeah, there's definitely overlap there. There is a response from Jerome, but we're waiting for him to enter, unfortunately, to type his response. Otherwise, the floor is open for any other questions. I think it's been a really fascinating and wide-ranging set of presentations. You know, I thought the topic of provenance would focus things down a little, but we've seen lots of really fascinating applications and interpretations of the topic as well. Everything from sort of smoothing the curve for non-technical users through to systems and platforms that enable provenance to be integrated into workflows and to travel with the data as it moves between, well, within systems and between systems as well. There's no silver bullet, it seems, for tackling provenance, and it's still a continued work in progress. And events like these, where we do get to come together and share ideas and thoughts, are really helpful. So I'd like to thank you all for contributing. We've got a response. Okay, bear with me 2 seconds. Thank you. High-tech approach of taking photos of phones. So yeah, in response to your question about the fact that we have libraries to facilitate access to books, the response is libraries are able to keep identical copies of books across a distributed network. This can be implemented with location agnostic digitally bound books also. So, with current digital data, only one location keeps the digital data. It's a single point of failure. Okay. I think, again, provenance and that last point, you know, the resilience of our data systems is something that is at the forefront of a lot of people's minds at the moment. I think it's something we all need to continue to work on and communicate and share knowledge about, because our data systems are more fragile than we realize. And I think, yeah, this community that's been brought together at this event, a best place to try and improve that situation. So if you join me in a round of applause for all our speakers, we'll draw this session to a close. Thank you all.